Ladies and gentlemen, on behalf of the Chemical Sciences section of the Hungarian Academy of Sciences, it is my greatest pleasure to welcome you to the second chemistry lectures of our academy. I will be your host tonight. My name is András Percel. I'm a professor of structural chemistry and biology at Eötvös Loránd University, Budapest. And at present, I am the chair of the Chemical Science section of our academy. The idea and concept of Christmas lecture is that in the middle of Christmas preparation, let's slow down. Let's dedicate some time and focus on important things such as spirit, science, and soul. This is the second year now that the COVID-19 pandemic that forced us to do more things via the internet. With all the unfortunate difficulties and struggles we had to face, let's notice the good part of this virtual space, the emerging and valuable opportunities brought to us, the great opportunity to enjoy the very best of sciences together, the opportunity of Christmas lectures. Let me welcome now our special and distinguished guest, Samuel H. Gellman, the Villas Research Professor of the Department of Chemistry, University of Wisconsin-Madison, USA, who is also the elected foreign member of the Hungarian Academy of Sciences. Before I give him the floor, please allow me to briefly introduce him, who many of us know well. Concerning his education, he started his university career by attending Harvard University in 1981, got his PhD at Columbia University in 1986, and became a postdoctoral fellow between 86 and 87 at the California Institute of Technology. Now, just very briefly about the science his group is interested in. Well, they're interested in the design, synthesis, and evaluation of new type of molecules intended to display interesting structures and useful function. In addition, they seek to understand how proteins, the most diverse class of biomolecules, perform their natural functions. Their efforts requires a wide range of experimental tools, including asymmetric organic synthesis, polymer synthesis, solid phase peptide synthesis, high-resolution NMR and crystallography analysis of molecular structures, protein expression, biochemical assays, and cell-based assays. His research program is highly collaborative and effective, which is, in terms of figures, is really outstanding. He has an H-index of 101 and citations over 44,000. Let me say a few words about perhaps one of his favorite topics, which we'll know more about tonight, foldovers. Nature teaches us that folded oligomers can be very powerful molecule machines, as exemplified by proteins and nucleic acid. Foldovers are unnatural oligomers that adopt compact, specific, and predictable shape. The foldomer approach represents a new strategy for design molecules that display specific functions. They are interested in developing foldomers that mimic the shape of natural peptides or proteins for biomedical applications, and in foldomers that adopt unprecedented shapes. They have learned a great deal about oligomers of beta amino acids, nicknamed as beta peptides, and oligomers containing both alpha and beta amino acid residues, alpha beta peptides, also called as chimera, which make these systems suitable for goal-oriented design efforts. They have shown that the properly designed helix-forming foldovers can disrupt protein-protein interactions and can be associated with viral infection and cancer. And I think that's probably the situation where we are concerning viral infections. And I just would like to read the title of his talk of tonight, Foldovers Extrapolating from Proteins. As I mentioned, Samuel, it's our greatest pleasure, and the floor is yours now.
Thank you. Thank you very much. I am deeply honored to be able to give this lecture. I'm very grateful for the invitation, but I deeply regret that I can't be with you this evening. I've always wanted to visit Hungary, but obviously that's not possible under these circumstances. So thank you very much for that introduction, which was very gracious and really sets up the lecture very well. I'd like to start by dedicating this lecture to the memory of Professor Ferenc Fulop, a major figure in the area in which I will, the scientific area that will be the topic of the lecture tonight. I have known Fulop via correspondence for many, many years. I was fortunate finally to meet him at a meeting in Paris a number of years ago. I'd hoped to meet him again in Budapest, but unfortunately this was not to be the case. Everybody in this field knows and was influenced by his research. So as was said in the introduction, the work is really inspired by thinking about the very powerful functions that we all know are manifested by folded sequence-specific biopolymers, specifically proteins and RNA. And as chemists, we understand that the building blocks of these two polymers are very, very different. And that difference, coupled with the similarity in terms of relationships among sequence, folding, and function, naturally lead to the question at the bottom of this slide: what about other building blocks? What about building blocks that evolution on Earth hasn't seemed to explore? As chemists, this is really a challenge and an opportunity for us, because it doesn't seem like only these two polymers will have these relationships among sequence, structure, and function. And so this is an endeavor that our lab undertook and many labs undertook in the 1990s. And so I'll talk a bit about structures, but mostly about functions this evening. So as Andres already mentioned, a lot of our work in this process of extrapolating from biopolymers really starts at the protein side, and then takes really just the smallest step away at the backbone level, moving from alpha amino acid residues to beta amino acid residues. So now between the carbonyl and the nitrogen, we have two carbons instead of one. Although we initially were exploring pure beta amino acid oligomers, or what we call beta peptides, now most of the work, everything I'll talk about tonight, is mixtures of alpha and beta, as was mentioned. So what we call alpha beta peptides. In the bottom, in the right box, you can see the kinds of beta amino acid substitutions that are possible. We can place the side chain, a proteinogenic side chain, one that mimics a proteinogenic residue, at either of two positions now, and we can do something that isn't possible with alpha amino acid residues. We can use a cyclic constraint to pre-organize the backbone. If you try to do this with an alpha amino acid residue, of course, you lose a hydrogen bonding site. You lose either NH or carbonyl. Proline, of course, is the example from proteinogenic amino acids. But when you have at least two carbons between the carbonyl and the nitrogen, now you can have the ring, the constraint, and maintain those hydrogen bonding sites, which you need for regular secondary structures. I should mention that although the goal is to create a tertiary structure, here's an enzyme tertiary structure on the left, that's a goal that's really pretty much still beyond the field, still for the future. What I'll focus on is secondary structure, and really specifically helical secondary structure. But I'll try to show you that even at that simpler level of structure relative to proteins or folded RNAs, there are interesting functional possibilities. When our lab became interested in this area in the 1990s, coincidentally, the Seebach lab also became very interested in beta peptides and alpha-beta combinations, and many groups have subsequently participated in this research. And I highlight that Thomas Martinek and Ferenc Fulop among many other groups, including ours, have been active in this area. I'll say a little bit more about that in a minute. There we go. So this slide simply illustrates some of the early findings from my lab. You can see how early this work was, initially with purely beta amino acids, where Dan Appel and Laurie Christensen found that when you change the cyclic constraint, you could change the nature of the helical secondary structure, the defining hydrogen bonding patterns, and therefore the global shape. And then later, Meg Schmidt, Alki Hayan, and Sikep Choi in my lab were able to find that if you began to combine alpha and beta residues, you could create a whole new family of defined folded structures. And these crystal structures that I show you, or these images of crystal structures, really represent multiple structures in each case, and then supporting NMR data as well. Early work was being carried out at the same time in Hungary. This is one of my all-time favorite Foldomer papers from Martinek and Philip and their co-workers, showing that not only composition and sequence of subunits, but also their stereochemistry could affect folding in a predictable way. Something I never imagined until I understood what was in this paper. I can say that this brilliant paper strongly influenced subsequent work in my group and in many other groups. So by now there's a whole zoo, a menagerie of different kinds of building blocks that have been explored. And I'm not going to go through all this, but if one moves from the upper left, where one sees gamma amino acid building blocks, sort of the next step out, and then just moves down the page, one moves to building blocks that are less and less familiar, more and more imaginative as they extrapolate from the starting point of proteins. And there are groups all around the world now studying these different types of foldovers. What I'd like to focus on, as I mentioned, is really function. Because although I love structure, and I think many other practitioners in this field love structure, I think we know that the broader community, doesn't just want to see pretty pictures. They want to know why one would build these molecules, what interesting activities could come from them. And so I'm going to talk about two areas of function this evening. The first is catalysis. And I think for an organic chemist, which is my background, this is probably the most demanding area, or at least perceived to be the queen of all functions that we find in the protein realm. And we know that enzymatic catalysis, I don't think we really understand how enzymes are as effective as they are at catalyzing reactions. But what we know is that enzymes bring to bear, through their folded structure, bring to bear on the substrate many different points of contact to achieve truly extraordinary reactivity. Here I show you a sort of a textbook picture of how a serine protease works. We know as chemists that an amide bond is exceedingly challenging to hydrolyze, and yet proteases, serine proteases, other types of proteases are very rapid in their ability to hydrolyze amide bonds and even to be very specific in terms of sequence. The serine proteases bring multiple functional groups to activate the carbonyl and activate the nucleophilic serine. The simple idea that I want to take from this picture, this textbook picture, is that if we can organize groups in space and bring them to bear on a substrate, then in principle, we can achieve catalysis. And the first step, I'm not going to show you how to create a serine protease, I don't know how to do that, but the first step is to organize two groups to act in concert, or what we would call bifunctional catalysis. So that's what I want to focus on. I will mention that there are a few reports from various groups over the last decade or so of Foldomer-based catalysis, some very nice chemistry from different groups. None of these, though, has addressed this issue of bifunctional catalysis that I'll talk about now. I also want to say that we derive a great deal of inspiration from the area of conventional peptide catalysts of organic reactions. I think the most eminent practitioner here is Scott Miller, but there are many others as well. In this field, though, I would say that the major focus has really been on stereoselectivity, often enantioselectivity. For us, the real issue is reactivity, because I think that's where enzymes are so special. So then the question becomes, well, what reactions should we try to catalyze? And our efforts were really inspired by a paper from the Pico lab in Finland a number of years ago. They deal with this crossed aldol reaction that you see at the top of this slide. This is a reaction that we teach to undergraduates. Formaldehyde can function only as an electrophile, obviously. Everybody's known for decades that pyrrolidine can catalyze this type of reaction. The thing that was special about this paper and that caught our attention was the demonstration that the pyrrolidine catalysis is second order in these reactions, which suggests that pyrrolidine is doing two things. It's activating the nucleophile as an enamine, and it's activating the formaldehyde as an aminium, so that both pyrrolidine rings are involved when the key carbon-carbon bond forms. And so that led Zeb Gerben, a former student in my lab, to wonder about Foldovers that contained this pyrrolidine beta amino acid that you see in the lower left. My lab had used this building block for years just to get beta peptides and alpha beta peptides to dissolve in water. But Zeb realized he could use Foldover scaffolds to try to arrange pairs of pyrrolidines in space to see if he could get them to work together to catalyze this cross-aldol reaction. He evaluated a number of different scaffolds, but the one that was most successful is the one that you see here. It has an alpha beta beta repeat, a 3-residue module that repeats. It contains this 5-membered ring residue for pre-organization. And the helix that's formed, you can see 2 different images here of a crystal structure, very regular helix that you see from the side view. But if you look down the helix axis, you can see that there's an almost perfect 3-residue per turn repeat. And that repeat means that 2 pyrrolidines with the so-called I to I+3 separation in terms of sequence should be aligned along one side of the helix, and they're about 5 angstroms apart. So this is the experiment then, or the type of experiment that Zeb conducted. He took a specific cross-aldol reaction with formaldehyde and hydrocinnamaldehyde, a specific set of conditions drawn from the Pico precedent. He then looked at relative initial rates of the cross-aldol using as his benchmark an alpha-beta peptide with 1 pyrrolidine residue, and then asked what happened as he introduced a second pyrrolidine with varying spacing along the sequence. You can see that if the 2 pyrrolidines are right next to one another in sequence, there's no effect on relative initial rate. That's because in the helical form that we expect, those 2 pyrrolidines are held apart, are splayed apart by the helical structure. But as you get the 2 pyrrolidines into just the right spacing that would allow them to be aligned along one side of the helical axis, now we see over 100-fold increase in relative initial rate. And then when we push them apart a little bit further, the catalysis largely dissipates. So this was a very gratifying result for us. It suggested that indeed you could use a simple helical Foldover scaffold to arrange 2 groups to achieve bifunctional catalysis. This work was published a few years ago, so I won't say more about the evidence, but every piece of evidence that Zeb acquired was consistent with this catalytic cycle in which both of the components, the formaldehyde, the hydrocinnamaldehyde, are activated on the Foldover scaffold, and the key carbon bond forming step occurs on the scaffold. Although we were excited about this result, we also noted in the paper that this best of our catalysts was not quite as good as pyrrolidine itself. Presumably these pyrrolidine residues in the beta peptide are deactivated by putting them into the peptide, perhaps because of the electron withdrawing groups, perhaps because of steric hindrance. But even if this peptide had been better than pyrrolidine, nobody who wanted to do this reaction would make this peptide. You'd always just pull pyrrolidine off your shelf because it's a cheap, readily available compound. So although this was an important demonstration for us in terms of the principle of bifunctional catalysis, this clearly wasn't useful from a synthetic perspective. And this consideration led us to start to wonder what might be useful from a synthetic perspective that a Foldover could do, that a small molecule could do. And we settled on the idea of making larger rings because we know that this is a bit of a challenging process for organic chemistry typically. And so we wondered whether we could form the dual activated species on the Foldover scaffold and then have that challenging carbon-carbon bond formation step occur on the scaffold and in that way catalyze the process. This 9-carbon dialdehyde, for example, could make a cyclooctene. It's hard to make 8-membered rings. And in fact, the catalyst didn't make the 8-membered ring. Fortunately, Zeb was very enterprising. He didn't limit himself to this study. He asked what happened if he took one of the amino groups and instead of putting it in a pyrrolidine, he just made a primary amine. He still didn't get this 8-membered ring, but now he started to see cyclodimers and cyclotrimers. This cyclodimer structure was elucidated by M.K. Andrews in my group. You can see this nice symmetrical 16-membered ring. So this was quite exciting to us. Here I show you the LC trace of this reaction after about 24 hours when, as far as we can tell, it's pretty much done. We can't see the starting material in the LC, so we're not exactly sure, but it's pretty close to done. So these two large peaks are the cyclodimers. There are 2 of them. I showed you the structure of one. I could guess the structure of the other, but we didn't elucidate it. There are also cyclotrimers, right? So these are even larger rings, 24-membered rings, and other things as well. It's kind of a messy reaction mixture. But if we just focus on the cyclodimers and we monitor them as a function of time, we see that the reaction for the Foldomer tops out at about 10 hours. But now if you look at a small molecule control, 1 secondary amine, 1 primary amine, we see that at 10 hours, this reaction is just barely getting started. So here is a reaction in which the Foldomer clearly has an advantage relative to the small molecules. So we decided to focus on this reactivity. We shifted at this point to a much more tractable reaction, a series of unimolecular cyclizations that would make large rings from a single dialdehyde. I show you an example that makes the same ring size we saw before, 16-membered ring. Now we're just going to talk about isolated yield, and you can see here the yield is excellent. So this catalyst is very effective, the one I showed you before. And what I love about this reaction is that the catalytic efficacy is exceedingly sensitive to the structure of the Foldomer, in the same way that we know that enzyme reactivity is very sensitive to mutations in the enzyme sequence. So I'll show you a few examples here. If you take the secondary and the primary amine, which for the ideal catalyst at the LCMS level gives essentially a quantitative yield of that 16-membered ring. If you just move the 2 groups a little bit closer together along the sequence, you essentially lose catalytic activity. As I showed you before, if you have 2 secondary amines, you see almost none of the product. If you have 2 primary amines, it's a little bit better, but still not nearly as good as the primary secondary amine dyad. If you simply change the configuration at one position, catalysis dramatically falls off, presumably because changing that configuration really destabilizes the helix. And then if you swap the positions of the 2 catalytic moieties, catalysis falls off quite a bit, showing that even though the alignment along one side might seem to be a strictly linear presentation, in fact, there is an orientation that varies depending on C-terminal versus N-terminal. And then the important control, if you use just the small molecule amines, very little of the product. So the Foldomer clearly wins out here. We actually used the Foldomer, Zeb used the Foldomer to build a small natural product. Much more challenging than making the ring actually was building this dialdehyde. But once Zeb had built it, he was able to achieve a very good yield of what is now a mixture of 2 regioisomers. So this starting dialdehyde, unlike the previous one, is unsymmetrical. So now we get both possible regioisomers. For the synthesis of robustol, that doesn't matter because then we completely erase the enal functionality to get to the final product. But this does highlight a potential limitation in this approach. Zeb also looked at a cyclization process with another unsymmetrical dialdehyde that would generate the core of another natural product called nasocycline A, and you see the result here. Again, a pretty good yield of this 18-membered ring, the kind of ring that would be very hard to form in other ways, but a mixture of 2 regioisomers. And it was thinking about this regioisomer issue that the next phase of this research developed, taken over now by my student M.K. Andrews. Because she realized that you could start, this is down below in the brackets, you see the dialdehyde that Zeb had used. But if you shift the positions of both of the aldehydes by 1 carbon, you could generate the dialdehyde shown at the top that should give the same basic core structure, But now one of the aldehydes is an aryl aldehyde, and so that can function only as an electrophile. So this should give only one regional isomer if the reaction occurs. The difficulty here is that we know, every organic chemist knows, that aryl aldehydes are less reactive than alkyl aldehydes. And so the challenge now is to find something that in this simple model reaction will favor the cross-aldol relative to the homoaldol of hydrocinnamaldehyde. Now, what you see down here is if you use the Foldomer that presents two secondary amines, neither of those aldol reactions is good, even though this was great with formaldehyde. If you use Zeb-Gervin's catalyst, one secondary, one primary, it's not very good at the crossed aldol, but it's quite good at the homoaldol, the thing we don't want. What MK discovered was that if you use two primary amines, you can now get, you still get significant crossed aldol, but you, there we are. You still get significant homoaldol, but now the crossed aldol wins. So this was quite a surprising discovery. I don't think there was anything in the very copious, very extensive literature on amine catalyzed reactions that would have allowed us to predict this, but it was an excellent result. The other thing that MK realized she could do with two primary amines is shift the functional groups to the alpha amino acid residues rather than the beta. And so she could use entirely cyclically constrained betas to fortify the helix. So our hypothesis now is that we're activating both aldehydes on the helical scaffold. We have 2D NMR data in my lab showed that this particular Foldomer is very nicely folded into the predicted helix by 2D NMR in isopropanol. And so now MK was set up to try the cyclization to the nostocycline core. And it works very well. You see an excellent yield obtained for this cyclization reaction with her catalyst. This situation, and in particular, the fact that the amino groups are both presented by alpha amino acid residues, now allowed MK to ask some very fundamental questions about pre-organization and the nature of the backbone and their relationship to catalysis. So for example, she could ask, what if we keep the alpha beta beta backbone, but we relax the cyclic constraints? We just go to these beta homoalanine residues. Well, you can see that this flexible alpha beta peptide is much less effective at cyclization. She could also ask, what if we go to a pure alpha amino acid backbone? Now the two possible helices are 310 and alpha. Alpha helix has about 3.5 residues per turn. A 310 helix has about 3 residues per turn. So MK looked at both spacings, I to I plus 3, which should be good for the 310 helix and a little bit less than a full turn of an alpha helix, or I to I plus 4, which should be just a little bit more than 1 alpha helical turn. But neither of these catalysts is any good. You can see they're just trace levels of product in these cases, clearly showing the advantage of the Foldomer scaffold relative to a conventional peptide scaffold. MK took on one more reaction, even more challenging. She tried to see if she could cyclodimerize the dialdehyde you see here on the left. Again, there's an aryl and an alkyl aldehyde. This is a more challenging process. It leads to a very interesting product, we hoped, that would be the core, they have the core of the cylindrocyclophane natural products. But the challenge here is that if the alkyl aldehyde reacts intermolecularly with another alkyl aldehyde, you generate this byproduct, which is stuck. It can't go anywhere. So that's a dead end. You have to get the aryl alkyl intermediate in order to be able to go onto the product. But MK's catalyst was great, as you can see, a very clean product. Here you see the LC trace, very clean product, very high yield of the cyclodimer. Whoops. In contrast, if you look at this flexible alpha beta peptide, at the same time period, 20 hours, mostly starting material, and although there is a little bit of product, you also get the intermediate, you get the byproduct, clearly a much less effective reaction. And same was true with the alpha peptides, although I don't show you this. So what this work shows, I would suggest, is that these Foldomer scaffolds can create very effective catalysts for very challenging reactions by placing two reactive groups in the proper spacing. And they can reveal subtleties in reactivity that I don't think one could discover in any other way, because I don't think there was anything in the literature that would allow us to have understood that these different dyads, 2 secondary amines versus 1 secondary and 1 primary versus 2 primary, would have these different aldol reactivity preferences. Only by seeing their action in coordination could we discover these reactivity patterns. I'd now like to shift gears to a second topic to finish the lecture, and this is a topic that was mentioned in the introduction, the idea of trying to block protein-protein interactions that are deleterious, that are associated with disease. Our goals will focus on helical segments that are involved in recognition processes, and the general thinking here, and this is general in a large field, is that if we could mimic the surface on the helix that's recognized by the partner protein, but change the backbone, then perhaps we could occupy the site, prevent the original alpha helix from occupying that site, but our molecule would not be recognized by proteases because of the backbone change, or at least not very well recognized, and therefore our molecule would not be degraded rapidly by proteases, which unfortunately is what happens to conventional peptides. I'm going to talk about antiviral strategies where this challenge is very evident. Here I introduce the problem by talking about the process by which the virus HIV infects cells. So many pathogenic viruses, including HIV, are enveloped, which means they have a lipid bilayer that surrounds them. And so the virus has to fuse its envelope, its membrane, with the host cell membrane before infection can occur, before the viral contents can enter the cell. So this process is orchestrated by a protein that sits on the surface of the virus. And for HIV, this protein is called GP41. It's a trimer, as is generally the case. And this cartoon in the upper left shows the state after the virus has recognized the cell and begun to engage the GP41 machinery. So at this point, the GP41 trimer adopts an extended form that is embedded both in the target cell membrane and in the viral envelope. This extended form is thought to be unstable, and it folds back on itself, as cartooned at the bottom, to form a compact structure where you see tan and blue helices bundled together. There's 6 of them, actually. This is called a 6-helix bundle intermediate. The formation of that compact intermediate brings the 2 membranes together. Ultimately, they fuse. Now the viral contents can enter the cell. Now infection occurs. Evidence for this 6-helix bundle intermediate came in the 1990s when a number of groups, first Peter Kim's group, showed that if you took the peptides that correspond to these long cylinders in the cartoon, these are long helical segments, and they will spontaneously assemble into a 6-helix bundle in the crystal. This understanding then led to the development of a drug called Fuvertide. It was approved almost 20 years ago. It essentially is the blue cylinder, the blue helix, and it's thought to catch the extended form of GP41 trimer in this extended form to block the folding back process. This drug is still effective, it's still used, but it's not been very successful because it has to be dosed at very, very high levels, and there are adverse immune responses that develop, and so patients, I'm told, really don't like it. We saw this as an opportunity to test the hypothesis that we could mimic the informational content encoded in the surface of a long alpha helix, like this long blue helix, but alter the backbone sufficiently that proteases would not be effective at degrading the peptide. This peptide in Fuvertide has a half-life of just a few minutes in the bloodstream because it's so rapidly degraded. Here I show you, or I'll summarize because this is old work, a strategy developed by Seth Warren, a former postdoc in my lab. Here, Seth used a strategy that was tuned to the periodicity of the alpha helix. He took sets of 7 alpha amino acid residues corresponding to 2 turns of an alpha helix, and at 2 of the 7 positions, he encoded a beta residue. The beta residue replacement is shown in cartoon below. In every case, he used the beta homolog, the beta 3 homolog. So same side chain as the alpha residue, but an extra carbon, extra methylene between carbonyl and the side chain bearing carbon. So the blue dots indicate where the beta homo is. The letters still indicate the side chain as they do with a conventional peptide. you can see that if we look at target engagement, I'll show you what this means on the next slide, the alpha sequence we're starting with here, which is already engineered to be a second generation AIDS drug, very potent binding to the target. This alpha beta peptide is much less effective. However, we are seeing some improvement in terms of proteolysis. We're using a very aggressive protease. Protease K cuts at many sites. You see we get about a 20-fold protection just by sprinkling betas in this way, but we've lost a lot of binding to the target. Now, our hypothesis was that the problem here is that the beta residue has one more flexible bond relative to an alpha. So with 11 replacements in the lower structure in this alpha beta peptide, there are 11 more flexible bonds. And so this should be a much harder conformational entropy challenge to fold into a helix. And we could test that hypothesis by incorporating these cyclic residues, which we knew were preorganized to promote an alpha helix-like conformation. And what you see is through 2 rounds of replacement, this is work from Seth and then a former student, Lisa Johnson, you can see the binding got better. And specifically, this is binding to an engineered protein that you see in gray in this crystal structure. This is 5 of the 6 helices engineered into a single protein. This is from Steve Harrison's lab. And then that presents the binding site for the 6th helix. And this lowest alpha beta peptide with the maximum number of tan dots, the maximum number of cyclic residues, you see fits very well into the appropriate groove. Now you can see the wisdom of Seth's placement of beta residues, because that placement causes the betas to line up in a stripe. The beta stripe of those blue and orange patches are all pointing out into solution, out toward us. The protein sees only alpha amino acid residues. And this is one of 8 crystal structures we have in the protein database. So this is a very reliable alpha helix mimicry strategy. You can see that the affinity got better as we added cyclic residues, and the protection from proteolysis got really a lot better, 300-fold better for this alpha beta peptide relative to the all alpha, even though this lowest peptide is still mostly alpha residues. Although the binding isn't quite as good as the all alpha sequence, it's still good enough to check viral activity. So we worked here with John Moore and Min Liu at Cornell Med School, and they were able to show against a range of viral strains for a cell-based infection model that this alpha beta peptide was quite potent at blocking. This is the all alpha sequence. The one with the cyclic residues was very good, comparable in many ways, in most cases, to the best alpha sequence at blocking viral infection. So this is work from quite a number of years ago. I'd like to tell you how we've carried this work forward in the last few slides. So that was really a model system for us because our collaborators told us that nobody was interested in a new peptide to treat HIV. And in any case, we're an academic lab, we're not a drug company. But our HIV collaborators introduced us to Ann Moscona and Matteo Parato at Columbia University. They worked on this virus, human parainfluenza virus 3, which is a very serious threat to children and to the elderly. It causes upper respiratory infections, significant cause of death for children, young children all over the world. There is no strategy to address this infection. Now, HPIV3 also uses a fusion mechanism. It's also an envelope virus like AIDS, completely different fusion protein. But the cartoon that you see here basically applies to HPIV3 as well as to HIV because this cartoon is so simple. And there is a 6 helix bundle here as well. Our collaborators already knew about this before we started to work with them. And they had actually developed some modified all alpha sequences that were improved in their potency for inhibition. You see one here, the second sequence, there are 2 letters in red. These are modifications that they had made to improve potency for blocking HPIV3 fusion. Here you see a crystal structure that my former postdoc Victor Outlaw and former student Dale Kreitler solved, showing this improved sequence from our collaborators with the core peptide from HPIV3. This 6 helix bundle looked exactly as it was supposed to look based on a structure of the post-fusion complex that had been published many years earlier. The reference is down here. The key point I wanted to highlight is the green part on the outer helices are mostly helical, but there is an extended segment. There is a non-helical segment. It's an irregular conformation, but a very specific one. So I've highlighted on the sequence that N-terminal segment that's not helical. Here you see now the result of a lot of work from Victor Outlaw and Ross Chiloa. Again, structural collaboration with Dale Kreitler. Here you see that that alpha-beta replacement strategy, 2 betas out of every 7 matching the heptide repeat of an alpha helix, you see that that works here as well. Mostly brown residues, mostly cyclic residues, but occasionally beta-3 homo residues, but no replacements in the non-helical portion, because that part we ran into trouble when we started to make replacements. Here's the co-crystal structure. Now the beta residues are the purple patches, looks just like the structure on the previous slide, no betas in the extended segment. And when you look down the helix, the axis of one of those green and purple helices, you see all the purple, all the beta residues are pointing out into solvent. They're not engaged in the interface between the green and the orange helices. Working on HBIV3 allowed us to go into animals, which is a very important advance, something we couldn't do with HIV. And so here you see results that were published earlier this year, again in a collaboration with Moscona and Parado, but now also with the newest lab at Ohio State. On the left, you see simply pharmacokinetics, what happens to the peptide 24 hours after intranasal delivery into cotton rat lungs. You see that the alpha peptide is essentially gone, presumably totally degraded by lung proteases. But the alpha beta peptide is still there. I should mention a very important point. These are not a simple peptide anymore. Our collaborators have learned that you need to attach a hydrophobic group, such as cholesterol, to one terminus for maximum potency. And so now we're dealing with lipopeptide versions. There's a short linker, flexible linker, and then the cholesterol. But again, we're comparing all alpha sequence versus our best alpha beta sequence in the lipopeptide format. So again, the alpha beta sticks around after 24 hours, the alpha doesn't. Now we can ask for efficacy. So this is now prophylactic dosing. So first we dose the peptide, then 24 hours later, we dose the virus, and then we wait 4 days and see what happens. You can see there's a lot of virus if there's no peptide. If there's the alpha peptide, there's some suppression, it's not very good. With the alpha beta, significantly better, presumably because the alpha beta peptide is not degraded as readily as the alpha peptide. So this was a very exciting finding for us, showing that we could be effective at blocking viral infection in vivo. And then finally, for the last couple of slides, the topic we've already discussed, the virus that's keeping us all apart now, SARS-CoV-2. This also is an envelope virus. It also uses a fusion protein that forms a critical 6-helix bundle intermediate, completely different protein from the other viruses I've talked about. But as we learned about this virus in February 2020, our collaboration with Moscona and Parado immediately pivoted to this target. Here you see on the left, well, on the very left, you see a cartoon that we've all seen too many times. There's a structure of the spike protein in its initial state. This is a cryo-EM structure published in 2020, sort of all balled up. And then a model for the extended state based on some other coronaviruses with the 6-helix bundle portion highlighted at the bottom. The next slide makes a critical comparison. So on the right, you see that 6-helix bundle structure, now a true crystal structure reported from a group in China in 2020. And this allows us to see that this 6-helix bundle term is really not quite valid anymore, because the outer portion, it does have a small helical segment in the middle, but it's actually a non-canonical helix. You can clearly see that there's a bend, but there are long extended segments on either end at the C-terminal and N-terminal portions. And I contrast that here, the situation with HIV, where it was all helix. I'm showing you a crystal structure of an alpha-beta peptide, different from the one I showed before, but alpha-beta, the blue and red patches are beta residues. Here's the HBIV3 structure I showed you before, different colors in the previous slide, but again, red and blue are beta. But as I showed you before, there is a small non-helical segment at one end. And here for the coronavirus, a lot of non-helical segment. So a much greater design challenge in this case. We initially simply asked the question, does the native alpha sequence inhibit infection? We were virtually certain the answer to this question was yes, because this sort of thing had already been shown for other coronaviruses, including the original SARS virus. Nevertheless, it was an important control, and I show you the results here. They were published over a year ago. This is the relevant peptide sequence with the cholesterol unit attached. And we're collaborating not only with Muscona and Parada, but also with the Bente lab, which had the facilities to work with live virus. This is an experiment using human lung tissue. You infect the human lung tissue once a day with the virus for 3 days in a row. After each infection, you wait 90 minutes, which is long enough, I'm told by my collaborators, for the virus to fully enter cells. Then you add the peptide. And then after those 3 days, you just wait and see what happens. And the virus has been engineered to express green fluorescent protein so you can see the virus when it grows out. So if you don't use any peptide inhibitor, at day 3, after the 3rd infection, you don't see anything. But day 4, you start to see viral growth, and by day 6, there's virus everywhere. So the virus is clearly spreading in the human lung tissue. But when you add the peptide, that suppresses the spread. You don't see anything, even at day 6. Okay, so this is a very fancy positive control. It shows that the native HRC segment is an effective inhibitor. Now, the challenge for us, and this is an ongoing challenge, is to modify the backbone to reduce resistance to proteolysis by retaining activity. I will say that our collaborators were able to show, this was published last spring, that if you take 2 copies of the native sequence and link them into a dimeric construct with a cholesterol, this is very potent at blocking transmission in ferrets. So this is a very exciting development showing real clinical potential. But this is the native sequence. So again, we hope to apply our backbone modification strategy in this context. The native sequence is very hard to work with in our hands, very insoluble. And so that was the first challenge we needed to address. So Victor Outlaw, my former postdoc working with the current postdoc SENU, developed the sequence that you see at the 3rd of the sequences at the top, what we just call peptide 1 here. It's got 8 modifications, the letters you see in red, intended to make it more water soluble. And this peptide, here's a co-crystal structure that Dale Kreitler solved. It looks exactly like the native sequence, even though there are 8 modifications. That's what we expected. This hasn't been published yet, but the PDB file is available. And most importantly, this soluble peptide is equally potent with the native sequence. So that was very gratifying. And then my last data slide, which doesn't seem to want to come up, just shows what, here we go, shows what we've wanted, where we are in our process. Because this structure is so, deviates so much from a classical alpha helix, right? The extended portions at each end, even the helix that's there is bent. We've had a very hard time making backbone modifications. And the structure you see in the middle of this slide with the blue and tan dots is the best we've been able to do. We can get some beta residues at either end. We can get them in the flexible linker. But even in the helical segment, we can't get the 2 out of 7 to work. We can only get 1 out of 7. So this sequence is equipotent with the all alpha sequence, and we're gearing up for the animal studies with this sequence. Maybe this will be enough beta substitution to give us the protection we need, but we're not very confident of that. From a design standpoint, our focus is on the question in the box at the lower right. What about other backbone modifications? This is really challenging us to move beyond beta amino acids in the context of this sort of bioengineering challenge. And it's an exciting challenge that we're eager to take up in the new year. But at least we've made some progress in this goal of protecting the backbone while retaining antiviral potency. And as I say, maybe this sequence will be good enough to be effective, to be carried forward in the fight against the coronavirus. So this is the science I wanted to tell you about today. Again, I come back to the question that motivated it by thinking about proteins and RNA in total. And so at the bottom of the slide, I'd like to suggest my conclusion, my general conclusion, that new backbones can be a source of new structures and new functional profiles, new combinations of functions that we don't see, that evolution hasn't called forth from her biopolymers, presumably because evolution didn't need that combination of functions, but a tremendous opportunity for chemists and chemistry, I think, as we move forward. I want to close by thanking the Hungarian Academy of Sciences for this tremendous honor, the opportunity to give this lecture and for membership, which I'm very proud of, and thank the outstanding coworkers in my group over many years who have carried out the research I told you about today. I mentioned their names as we went along. Of course, this work depended very heavily on collaborators, whose names I mentioned as we went along. I show you a picture of my group that's now a few years old. This was pre-COVID picture. The members of the current group are listed here, but the central privilege of my career is working with these outstanding young people, this ever-renewing stream of outstanding young people. So again, thank you very much for your attention and for the honor of giving this lecture.
Congratulations. Can you hear me?
I can.
Yeah, that was outstanding. I mean, of course, as expected, but this is really fantastic. So congratulations on all these nice data and all the achievements. And I think this was very nice and indeed, I wouldn't say easy to follow, but clear to follow. So I will have once it's will be on and have time to listen it again. But I think that it was really a very nice combination of details and an overview. So thanks once again.
Thank you.
Now, in the meantime, I mean, in principle, we could hopefully have people to be brave and courageous to ask questions. I'd like to start by saying a pretty general question regarding catalysis. I mean, the first part of your talk. So, how do you see? I mean, the way how these things has evolved and how you've shown that in which area this is useful and for what area to move ahead. So how do you see the future of basically Foldover, I would say Foldover catalysis? So I think it's green, it's biodegradable. So in many aspects, I think it's very promising. But on the other hand, as you mentioned, it's a bit complicated in terms of structure.
Well, so I would say two things. The first thing I would say is, actually, building those short alpha beta peptides is really not complicated. I know that most organic chemists view it that way, but my students using a microwave, just a hand microwave, nothing fancy, can make the peptide in a day. It's no problem at all. So
exactly.
Yeah, I mean, when you look at what people do to make these fancy phosphine ligands, it's very easy chemistry. It's very straightforward. But I think actually you're asking a much more profound and very important question. So what are the interesting applications? What are the challenges here? And we struggle with these things ourselves. On the one hand, I would love to build beyond bifunctional catalysis. We could think about, and we are starting to try to organize three things, see if we can get trifunctional catalysis. But really, what we'd love to be able to do is start to create more of a pocket. And that is more challenging with Foldovers because mostly our control is at the level of secondary structure. But then we also want to understand where can we achieve maximum effect through harnessing these scaffolds. And actually in my group, students have convinced themselves that photocatalysis is an area that is potentially very useful because of the many of the photocatalysts, especially for those that are not based so much on electron transfer, but energy transfer, have very short half-lives. And so if you could get a photocatalytic group on the Foldover and a substrate, you could potentially have very powerful and efficient effects there. This is a good example of what happens in my group. I know very little about this field, and so the students are leading me, and we'll see how they do. I'm cheering them on.
Yeah, absolutely. Yeah. One aspect of when, you're absolutely right. I mean, you use a microwave-assisted peptide, solid-phase peptide synthesis or flow chemistry. I mean, my point is not that making it would be too complicated, but in general, how to say that, and that concludes to my question that how do you design something? So you do some kind of how to say that, molecular graphics or some kind of a computational tool when you design certain things which would be more appropriate than the current one you use, or you have any, gut instincts, or how does it work in lab?
So this is also a great question. I have no doubt that the day will come when computational tools will be very useful in this field. As far as I can tell, that day has not come yet. And we don't use computational tools. When we initially developed, when we were initially using the crossed aldol with formaldehyde, Zeb Gervin was using it really as a screen to find the scaffold that was best. He screened through multiple scaffolds. He simply took the structures that we knew about, that we'd already characterized, and did that trick of multiple positionings of pyrrolidines to find the best one. So it was empirical based on structures we knew. So far, we're really trying to build from our knowledge base. So it's rational in that sense, but we're not designing anything new because I don't think we know how to design anything new yet. I wish we did.
I see. Because I mean, as you pointed out in the last two examples with coronavirus and with HIV, I mean, then we really targeting, big systems, and once you have these large, evolved systems, then it's really a crucial thing to really be able to place it in and me as an NMR person dealing with dynamics of these macromolecules. I mean, this is just amazing how successful you are. I mean, that slide on the lung infection and the peptide working. I mean, this is just congratulations. This is outstanding. This is wonderful.
Thank you. Yeah. Well, I was certainly excited to see that.
All right. Somebody's asking me, doesn't have a microphone, why did evolution at the beginning prefer alpha peptidoproteins rather than beta and alpha? So what do you think? This looks like, I know this is I think this is a good question. So we know that from asteroids and others, beta amino acids are available. So during chemical evolution of the building blocks, I mean, alphas are not better or superior than betas. Nevertheless, beta seems to be excluded, at least from the conventional type of peptide and protein making. Of course, we know that in antibiotics, all these things, we have the building blocks built in. But the question would address to you would basically help us to know more about your view on how this alpha to beta preference was shifted during evolutions. Why don't we have chimera? Why don't we have basically beta amino acid, proteins made fully of beta amino acids?
So, as you say, this is a wonderful question. I think there are two questions here, really. One is, why not pure beta? Another is, and I think the questioner was specific about, why not mixed alpha beta? And of course, it's purely speculation what I'm about to say, but I see two different kinds of answers. For pure beta, I view the biochemistry that we have today as sort of a result of a combination of thermodynamics and kinetics, in the sense that the system was trying to optimize itself evolutionarily, but at a certain point it became committed to something. This is the kinetics part. It became committed to a certain path, and then there was no going back. And so, to me, the question of why alpha and not beta, maybe just the alpha system, there were more alphas around, and so the alpha system developed first, and the betas never had a chance. I don't see an obvious reason why alphas or betas are better relative to one another. But I think the answer for the mixed backbones is a little bit different, because there, if you think about even all biology, one of the many wondrous aspects of biology at the molecular level is the speed and fidelity of heteropolymer synthesis. And so right, we have the ribosome, we have polymerases. The ribosome, I think, could not be as efficient as it is if it had to make both alpha and beta bonds. Because to be really efficient for either one, you have to have everything positioned in just the right way for the key amide bond forming step. And the ribosome doesn't do a very good job with beta amino acids. A number of groups have shown this. A few groups are starting to get beta amino acids into proteins, but it's still not very efficient. Presumably, the biology couldn't optimize that, this is my view, couldn't optimize heteropolymer synthesis with a mixed backbone. And so at a certain point, it just had to be committed to one. That's my personal hypothesis.
I was just pointing one interesting point, which goes back to history. Once we had, a long time ago, joined the same dinner in Oxford, St. John's College. And after that, we had this discussion about the overall stability of having amides separated by 2 or 3 flexible bonds. Because in alpha, you have basically phi and psi, like in a beta amino acids, it's in principle 3. And then one thing you mentioned and was very convincing to me, and that's I'm still quoting that, is that basically that's one issue, flexibility as an unfolded system. And I bump into this problem when we work with unstructured proteins several times, but when it's folded and builds up non-covalent interactions and get a nice folded things, that's another issue. And for some reason, it looks to me that beta amino acids can be, even without cyclic rings, can somehow end up being more stable, so thermodynamically, in terms of delta G, something more stable than alpha. And as we agreed on that years ago, is that if something is too stable, that's in terms of evolution could be a problem. So when you design a Foldomer and you optimize on delta G by optimizing the interaction, that's good. But when you try to evolve things, an overstabilization may not be so good in terms of perfection. But I would be very happy if you would still comment on that.
Sure. Well, I think these considerations are absolutely correct. And we have tended in my group, and I may have said this to you many years ago, we've always tried to design the maximum conformational stability because we know the easiest thing for a chemist to do is to make things less stable. We always know how to do that, conformationally, chemically, whatever. So I think it's really uncertain in terms of conformational stability, whether the acyclic beta amino acids, how they compare with alphas. You know, early on, Seebach claimed that the acyclic beta amino acids formed more stable helices than alpha amino acid peptides. I don't think this is correct. And the evidence at the time was very slim, but somehow that hypothesis has been propagated. And I think there are clear counterpoints. So I think beta amino acids are as flexible as one would need them to be for exactly what you're discussing, which is the need to maintain dynamics for sophisticated function. And I think it's a very important point.
Actually, going back to my former question, I don't know, just thinking about it some more. It is also possible that, once you became committed to alpha amino acids and that the tRNA synthetases evolved through gene duplication, then it became very different to evolve active sites that would accommodate amino acids. So you're really locked in.
Yes.
Yeah.
I completely agree. That's what I was trying to indicate in an unclear way when I talked about kinetics. You're sort of kinetically trapped in the alpha world, and you sort of can't. That's the way I was thinking about it.
Yeah. I agree.
Yeah, that's correct. I don't see people too shy, probably, or polite or whatever, to ask you more questions. So,
may I have a question?
Yes, Peter.
Yeah. Would it be a good strategy to make the backbone more rigid to increase reorganization for the target?
This is always a profound question because I think what organic chemists have learned through decades of post-gas chemistry and this kind of molecular design is that the more as you rigidify, things can get better and better in terms of binding if you're, let's say, lowering the entropy cost of binding. But as soon as the rigidification freezes out what the specific structure you need, then you're dead. And we don't have exceedingly fine control of structure. So I think in general, rigidification should improve binding as long as the motions that we're excluding when we rigidify are not, or the shapes are not the things we need. And also going back to Andres' point, to the extent that if binding requires a reorganization or function requires multiple states, their rigidification can be a problem too. So have I answered your question, or maybe I misunderstood?
It was excellent. Yeah, I agree. Yeah, thank you very much.
Okay, thank you.
Again, another question from a guy having no microphone. I'm trying to do my best. So his point is that would you comment on the possibility of basically using, I think the question pointed on, using bacterial system, using the conventional ribosomal expression system, but hooking instead of alpha beta amino acids to tRNA. And so that could be a feasible system to make basically chimera peptides. So what is your vision of that?
So I'll say that this is my dream, right? I dream of the time when we could use a technique like phage display to screen not only alpha amino acid side chains, but also backbone. I doubt I'll live to see that achievement, but I would love to see it. There are a few labs around the world that are working on this problem. The one that has made the most progress is the Suga lab in Japan. They've had a number of nice papers showing that beta amino acids can be incorporated into cyclic peptides. A few other labs have reported things that are may not be completely reproducible, but I still think what we see is that a number of labs recognize this fascinating challenge, but so far progress is quite limited, so there's a long way to go. And I think it's an open question whether we'll ever get to the point that we could do the kinds of experiments that the questioner wonders about, that I also dream about. I contacted Suga in Japan, who I regard as the leader in this field, last year to ask him if he could make 100 micrograms of an 84 residue peptide with one beta amino acid. Because I told him, if we have 100 micrograms, we can do an experiment that will give us a very exciting outcome, which, however it turns out, we'll have a wonderful paper together. Just 100 micrograms is all I need. And he wrote back to me and he said, well, if I have $250,000 in a year, maybe I could make 100 micrograms. But he said, I don't have either of those. So that to me just said that this challenge is still very, very far in the future. But it's a very exciting one. I tend to be a pessimist about progress. I'd love to be wrong with what I just said. I'd love to see these tools develop.
I see. Well, yeah, I mean, you're absolutely right. That's probably one of the goals to achieve in the near future. Okay, so to conclude, I have here not only one, but 2 diplomas. So I would say like 2 in 1. But that's pretty logical because as you are the elected foreign members of our Academy of Sciences, so you deserve 1 diploma, which of course will be shipped to you. And I won't read this because one part is made in Latin. You can Google it. The other one is made in Hungarian. So that's from something which is, of course, not so obvious. But the other one is in recognition of this current Christmas lecture, which then I'm going to read, of course, not the Latin, but the English part of it. So the chemical science section of the Hungarian Academy of Sciences is pleased to present a certificate of appreciation to the internationally renowned scientist Samuel H. Gellman, University of Wisconsin-Madison, in recognition of his outstanding contribution as a Christmas lecturer to advanced chemical sciences in Hungary. So with that, I'd really like to thank you to join us and to give this outstanding lecture. And I'm very much hoping that you'll be successful enough, one way or another, to find some cure against this epidemic. And once it's over, welcoming you in. It's nice to have you in a virtual space, but it would be even much better to have you in a real world here. So thank you again for giving this outstanding lecture. Thank you for entertaining us in the last more than an hour. And I wish you a very, very happy Christmas and a nice New Year, and hope to see you here in Hungary, maybe next year, maybe the year after.
Well, thank you so much for this honor. I really appreciate it, the honor both of the lecture and membership in the Academy. And I also hope I can see you in person, both in the US and in Hungary, which I've never visited. So thank you very much. And I wish you the best, all of you, in the holidays and the new year.
Thank you again, Samuel.
Okay,
Thanks.
have a good evening.
Thanks for everything. Thanks.
Okay,
Thanks.
thank you. Bye-bye.
Bye-bye. Take care.
You too.